A very good morning. My name is Darian Raja, matrix number 180673. So this video will be on my LAX number 6, which is about storage disease. So the type of storage disease that I'll be talking about today will be the Niemann-Pick disease in kittens, or also known as sphingomyelin lipidosis. So in this video, I'll be talking or summarizing what Niemann-Pick disease is, what causes this disease, what are the clinical signs, and what is the diagnosis and also the prognosis. So basically, Niemann-Pick disease is a type of disease that is caused by the buildup of sphingomyelins or cholesterols in the tissue. And what causes this accumulation or build-up in the tissue is due to the lack of sphingomyelinase, which is a type of enzyme that breaks down this sphingomyelin. When there is a lack of this enzyme, there will be an accumulation of sphingomyelin within the cells of nervous systems and also organs such as the liver, spleen, kidney, lungs, and intestines. Now, when there is an accumulation of this sphingomyelin, it will show certain clinical signs such as ataxia, which is uncoordinated movements, head tremors, loss of balance, splayed legs, splenomegaly, which is the enlargement of the spleen, hepatomegaly, which is the enlargement of the liver, and also progressive paralysis. Now, this disease mainly attacks the nervous system. It causes nervous system dysfunction, especially in the cerebellar dysfunction and also cerebrocortical dysfunction. And these clinical signs start to appear within the first few weeks of age, and it progresses and becomes worse as the animals gets older. And what are the diagnoses for this type of disease? Basically, the diagnosis will be based on the clinical signs, histopathology, and also biochemical evidence of reduced sphingomyelinase enzymes activity. Tissue accumulation of sphingomyelin is also done to see the amount of sphingomyelin in the tissue. And the prognosis for this disease is very poor. There's a high mortality rate in which kittens with this kittens with this Niemann-Pick disease will actually not survive, and most of them die within the first few weeks of this disease. So that is basically a brief summary on what Niemann-Pick disease is. Thank you.
Very good morning. My name is Hokaitong, matrix number 179019, and today I'm here to share about our case study, which is Niemann-Pick disease in kittens. First of all, you have to know that this disease is also known as NPD or sphingomyelin lipidosis. So this NPD actually is a lysosomal storage disease, which is caused by the autosomal recessive hereditary disease. And due to the lack of enzymes sphingomyelinase, so accumulation of sphingomyelin within the cells of nervous system and organs will be occur. So there are several clinical signs that we can detect its NPD disease. First, it's ataxia, which is known as uncoordinated walking, hypermetria, high stepping walk, heat tremors, and loss of balance and splayed legs. Um, actually, there's um, there's some diagnosis to determine whether the disease is NPD. And for your extra information, NPD have 5 types, which are A, B, C, D, and E. But in feline, there's only 2 types of NPD. There is NPD type A and NPD type C. A is in acute neuropathic forms, and in C it's chronic neuropathic form. So the diagnosis of this disease is basically rely on demonstrating enzyme deficiency in the tissues such as liver, brain, and also skin fibroblasts. So there's usually the scientists usually they will diagnosed by skin biopsy, where the sample is taken and used to analyze how the skin cells grows and how they move and also store the cholesterol. The second method is DNA testing. They will look for the genes that will carry or causing this type C NPD. So there's no treatment for this NPD disease, but but there's some in humans and also mice. In mice, treatment of n-butyldeoxynigerimycin will use, and this shows some delay in development of clinical signs, but there's no helps. So in human, they will use miglustate medication, which is an enzyme inhibitor to prevent the body from produce too many fatty acids. So I think that's all I learned from my case study. Thank you.
Okay, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Amir Shahid bin Dola and my matrix number is 180599. Okay, today I'm going to do the last case study, which is about storage diseases. Okay, our group has been assigned with Niemann-Pick diseases in kitten. Before we talk about Niemann-Pick diseases, I would like to talk about glycogen storage diseases. Okay, glycogen storage disorder are group of inherited disease. They result from a problem with one of the protein known as enzyme involved in the conversion of glucose to glycogen or the breakdown of glycogen back into glucose. They mostly tend to affect your liver and muscle. Most are diagnosed when they are child between 1 to 5 years. Symptoms include weakness, tiredness, and low blood sugar level. Newer treatment possibilities provide hope for an improved outlook. Okay. Glycogen. What is glycogen? Your body normally needs the sugar, glucose, to provide energy so that your cell and organ can function normally. Glucose is stored in your muscle and liver as a substance called glycogen. Glycogen is essential made up of a number of glucose units joined together. Various different proteins and enzymes help to convert glucose back to glycogen for energy storage. Okay. In Niemann-Pick diseases in feline, the disease is called Niemann-Pick disease type C. It's an autosomal recessive neurocervical lysosomal storage disorder in which cholesterol lipidosis result from defective intracellular transports of unspecified cholesterol. Okay. In this storage, kitten symptom for kitten is such as trembling when they walk, and then enlarge of liver and pancreas. And then there's one more is the pattern walks. They tend to lift their legs higher than usual, caused by these Niemann-Pick diseases. Okay, in Niemann-Pick diseases nowadays does not have treatment because it's very rare to occur in a kitten, so not many people nowadays try to go further about the treatment of these diseases. Okay, that's all from me. Thank you.
Good morning, my name is Norhani binti Abdul Mubin, metric number 180702, and today I'll be talking about our last LAX activity, which is about storage diseases. So storage diseases are a group of genetic disease associated with the failure of metabolic pathway, where there is an accumulation of substances within the lysosomal system, such as the lipid or protein within the tissues. hence it causes impaired cellular function. So there are two commonly reported storage diseases in veterinary medicine, such as the lysosomal and glycogen storage disease. So for our group, we were assigned to do on one storage disease, which is the Niemann-Pick disease. For feline, it is actually this Niemann-Pick disease is also known as sphingomyelinosis, or also known as feline Niemann-Pick type C. Actually, in humans there are 5 types of Niemann-Pick disease, which is type A, B, C, D, and E. Whereas in feline, there are actually 2 most common ones, which are type A and type C. Type A is the acute neuropathic form, whereas type C is the chronic neuropathic form. So this disease is actually the lysosomal storage disease. It is an autosomal recessive hereditary disease due to lack of the enzymes sphingomyelinase that results in accumulation of sphingomyelin cells of the nervous system, and also organs such as the liver, spleen, kidneys, lungs, and intestine. So this disease occurs most commonly in breeds such as the Siamese cats and also the domestic short-hair breeds. So for the clinical signs of this disease is it shows apparent from 3 months of age. The signs are such as ataxia, which leads to uncoordinated walking, hypermetria, head tremors, loss of balance, and also splayed legs. For some cats, they also may show some symptoms such as stereotypic chewing, and also their liver and spleen may be palpably enlarged. And when it becomes worse, progressive paralysis may develop, where most cats they die before 1 year of age. So this disease, it is the neurological deterioration, where it will result in delayed of mental and motor development and also premature death. Hence, So for this disease, there are no treatment that are be found. So the prognosis for this disease is poor because there are no treatment that they are still doing research for this disease actually. So most cats or kittens that encounter this disease may eventually die due to poor prognosis. So that is all from me. Thank you.
Hello everybody, my name is Tango Jaying and my matrix number is 180613. I'm from LAX group 16 and our group has been assigned to discuss about one of the storage disease, which is the Niemann-Pick disease in kittens. So what is exactly Niemann-Pick disease? It is actually a hereditary disorder where a deficiency in the enzyme sphingomyelinase. This causes sphingomyelin to accumulate in the brain and several other vital organs, which will eventually cause death of the kittens. So, sphingomyelin. What is sphingomyelin actually? Sphingomyelin, SPH, is actually a particular type of lipid that makes up the majority of the fat content of cell membranes. SPH structures consist of a polar group, a sphingosine, and a fatty acid. The polar group is made up of a molecule of either phosphoethanolamine or phosphocholine. This is connected to the sphingosine, which is an amino alcohol molecules with a total of 18 carbon atoms. The fatty acid part of the structure is usually saturated and it is connected directly to the sphingosine. Most lipids that form part of the structure of cell membranes are derived from glycogens. However, SPH differs differs from most cell membranes lipids because it is usually built from the amino acid serine and the fatty acid palmitate. The sphingomyelin synthase, or known as the sphingomyelinase, it is the main enzyme that is responsible for this chemical reaction. These molecules form a vital building block in the structure of cell membranes. SPH and the cholesterol are often discovered in high concentrations together in a certain region of the cell membrane, leading to the hypothesis that the metabolism of these two substances may be interrelated. Another important function appears to be the signal transmission in the nerve cells. This is in keeping with the observation that the myelin sheath has a chemical makeup with a very high sphingomyelin ratio. In addition to the concentration found in the nerve tissue, sphingomyelin is also a particular important component of red blood cells and certain other cells in the mammalian's eyes. So that's all about the sphingomyelin, and I'll end my part here. Thank you.